Okay. Yeah. So good day, everyone. I'm Jingwei, a senior research fellow from NUS, and here with me today is Russell, who is currently doing his master, and we are both from Profpo Engineering Biology Lab. So today we are going to introduce you to this BMSS software package, which is an automated biomodel selection system for genetic circuit design. So here is an outline for our presentation today. So to start with, we will give a brief introduction to cover the purposes and the challenges of doing the model selection. And next, we will introduce you to an overview of the what BMSS offer. And next, we will proceed to go through the model selection, which is the key feature offered by BMSS. And we will first start with a brief introduction of the case study, followed by a demonstration using the graphical user interface and also the Python script. After the model selection, we will cover the part for the model analysis using the sensitivity analysis and also the identifiability analysis using Python script as well. And next part, one of the important part for this BMSS system is database driven. So we will walk you through like how to actually handle the database to retrieve and store the models. And lastly, it will be focusing on we will focus on how to import and export the standard file in the form of Sbml file to enhance the interoperability among the software. So to begin with, in synthetic biology, we do modeling to help us to gain more insight into the behavior of the system and also the genetic circuit behavior. And it can also help to optimize the circuit performance. So show here is a simple inducible promoter system where we actually use to drive the expression of the red fluorescence protein. So this type of genetic circuit can be actually modeled using the kinetic modeling in the form of ordinary differential equation. So shown here are the two simple equations that we normally use to describe the transcriptional part to produce the mRNA and the translational part to generate the fluorescence protein. So this term is the part where it is actually dependent on the concentration of the inducer. So by just using this simple model, we will be able to generate this kind of like expression profile as shown here under different inducer concentration. So for this kind of case, the underlying assumption for this expression profile is that this is actually under a constant induction. So, but for some of the system, we realize that not all the inducible promoter system will express this type of expression profile. So there are some cases where the expression will start to increase and then reach a maximum expression and then start to go down again. And even the same expression trend will be can be observed for different inducer concentration. So this trend is speculated to be due to the inducer degradation or in terms of the binding instability. So for the last figure will be what we can see here is that there is actually a significant delay in the expression profile compared to what we commonly observe. So this delay induction could be actually due to the active transport of the inducer. So to actually capture all these different expression profile, we will normally have to go through a repeated modeling cycle, start with to identify the purpose of developing the model, what we expect the model can offer, and then we will proceed to derive the model structure. In this case, it will be in the form of ordinary differential equation that carries the different hypotheses. Next, we will actually move on to estimate the parameter value by fitting the model to the experimental data. Model analysis can then be performed to help us to identify the sensitive parameter that can be used for further tuning. And then this process we will move on to the optimization step to repeat this whole modeling cycle until we manage to identify the right model that can actually well capture the experimental profile. as we know, this modeling cycle can be difficult, but yet tedious. So it's important to expedite the process and also avoid the reinventing the wheel. So aside from there, we also observe that for different genetic circuit with different complexity, it's likely that they are going to exhibit different expression profile. However, interestingly, we observed that some of the hypotheses underlying the model are actually conserved across the similar regulatory system. So to avoid the repeated effort, so it's important to have a library of models stored in the model bank to allow the model to enable the model to be reused in the future model selection process instead of starting from scratch. So to address those challenges, here we come up with the BMSS system, which is a BioModel selection system. So this system can actually take in the experimental data and also the configuration file that encode the model structure and also the setting required to implement the model. So one of the more importantly, this system is actually database driven, where we can actually retrieve the model or store the model into the database. So after this is being done, we can then proceed to assess the different features provided by this system. So here we have the simulation, the curve fitting, and also the trace analysis, which allows one to determine whether the parameters are able to converge properly. And then the most important feature is definitely the model selection part, where we can compare like multiple models to identify the right model that can well capture the experimental data trend. However, how do we actually select the right model here? So for this system, we actually based on the Archive information criteria known as AIC. So this AIC is actually try to strike a balance between the goodness of fit and the model complexity to avoid the overfitting. So by using this AIC, we can actually get a ranking table to rank the performance of the different model, and from here we can identify the best model that can actually well capture the experimental data trend with less complexity. Next, we can actually proceed to also perform the sensitivity analysis to identify the sensitive parameter that can be used for parameter tuning, and also the identifiability analysis to highlight those unobservable states or unidentifiable parameters. So as we know, these are some of the key regulatory systems that are commonly used to regulate the gene expression in synthetic biology. So here is the part for the inducible promoter system, and this is the constitutive promoter system that is active in all the circumstances. And this is the logic gate system, where we have the NOT gate, AND gate, OR gate, etc. So for today demonstration, we are going to use OR gate as a case study. So in this case, we are trying to express the rate fluorescence protein driven by two inducible promoter system. And the first one is the P-type promoter induced by ATC, and the second one is the P-back promoter induced by arabinose. So this plasmid was transformed into the E. coli top 10 and culture in the minimal M9 media and characterized for a duration of like 12 hours. So what we show here is actually the characterization data obtained from the microplate reader. So the first input is actually the present, represent the presence of the arabinose, and the second input is the presence of ATC. So this blue line here is the result obtained in the absence of both inducer, and the red line is the result obtained in the presence of both inducer. And the green line is when we added the arabinose inducer, and the orange line is the result after adding the ETC. So this is actually the fluorescence per OD data. So this bar chart actually shows the maximum expression level under these 4 different states. So in order to perform the model selection to capture this trend, so here for this demonstration purpose, we only focus on three different type of model that carries different hypotheses. So in real case, we have tested these data using about 9 models that with different hypotheses. But for this demonstration purpose, yeah, due to computational time, we only focus on we only try to demo 3 examples. So for this first example, it is actually just a commonly used OR-gate model. So we assume that both the inducible promoter are under the constant induction, and the third part of the equation is actually is just to take into account the OR-gate operation. So for this second model, is actually we try to use a model with like opposite hypotheses. So for example, this case, we assume that this green line is actually under the inducer degradation trend, and this orange line is under delay induction. Whereas for the last model, we actually is actually expected to be the correct model. So this part, we have the delay activation model to capture this green line, and this part is for the inducer degradation trend. And aside from that, we also take into account the resource competition phenomenon, because by looking at this expression level, we observe that when in the presence of both inducer, the expression level is slightly lower compared to the summation of the expression level in the presence of only a single inducer. So with this, we speculated that it could be actually due to the presence of the resource competition phenomenon. So here is the workflow for the model selection process. So first, we will start with like import the experimental data file, and then we proceed to choose the models for the model selection process. And here we can actually either choose the model stored from in the originally stored in the database, or we can actually insert a new model that is encoded inside the configuration file. Next, we can proceed to choose the optimizer to be used for the curve fitting purpose. And then we also need to update the setting file. So here we have the initial value for the different states, and the initial guess for the different parameters, and also the parameter bounds that are required for the optimization process. And then for some of the cases, we can also fix some of the parameter to avoid those parameters from running through the optimization step. And then the last one is actually the time span for the simulation. So once those things is done, we can actually proceed to run the model selection. So yeah, this slide, I will actually walk you through a demonstration using this recorded video. So as we know, first we actually need to insert the experimental data file. So by clicking on the button here, the import file, we can actually import the experimental data file from our folder. Yeah, so this is the data file for the OR logic gate. So when we select the file, we just click on open, and then what you can see here is that the file link will be displayed in this column here. And then we just click on the upload file, upload data to upload the file. So next, we actually need to select the data file number. So here, because we only have one data file, so we just click on choose one. And then after that, we also need to insert the data type. So in this case, this data type is actually the fluorescence per OD data. So once we inserted the data type, we can just click on add to the system. So after that, we can actually proceed to visualize the content inside the Sbml data file. We can just by using a cursor and just click on the link, and all the Sbml data file will be displayed in this table. So what we can see here is that these 4 columns are the result, the mean value for the 4 different input, and followed by their standard deviation for the 4 different input as well. So we can then scroll down to look at the Sbml data point at different time point. So once everything is done, we can just click on the OK. And then a warning, a message box will actually pop up to show that your data file with this file name and the type was uploaded successfully. So the next part is actually to select the model. So we can either upload a new model or choose model. So, but for this case study, we assume that all the model already stored inside the database. So by clicking on the choose model function, choose model button, we can actually view a list of the model available in the database. So we just select the model, the OR gate model that I mentioned earlier. And then the second is the inducer degradation with delay. And the last one is the delay with integration followed by the resource competition. So once we select the model, we can just click OK, and then it will show that this model will upload properly. So the next step is actually to select the optimization method. So you can just click on this drop-down menu. It will actually show a list of different global optimizer. So for this case, we will be using the simulated annealing method. And then we can just click on the proceed to settings. From here, we can see that a file has been generated automatically under this name, the setting template name. And then you just click on the OK button. And then this setting, upload setting file window will pop up. Then we can actually go to proceed to visualize what is inside the setting template file. So as you can see here, this is the first model. And then followed by all the other different settings that originally available in the database, followed by the second model that we just selected and with their setting. And this one is the last model and with their setting as well. So for this case, we still need to modify the initial value and also modify the parameters. So for the modify init part, for the model 2 and 3, we actually need to turn on the inducer state. So in this case, we are going to set to 1. This is a normalized value. And then the 0, and 2 index is actually represent the index for the 2 inducer states. So next, we proceed to modify the parameters. So in this case, we have the 4 scenarios, the 0, 0, 0, 1, 1, 0, and 1, 1. So the -2 and -1 index is actually represent the state 1 and state 2, that whether we want to turn the promoter system on or off. But under certain conditions, we may not need to modify the initial value or the parameter value. If that's the case, we can just click on this button. Yeah. But for this case, we are not going to click on the here because we are going to modify this. And then we just click choose model. And here is to upload your setting template file. So, and then you just click on the open. So currently, we already kickstart the model selection process. So we just have to wait for a few seconds for the result to pop up. So this is the result window. So what we can see here is that the first section represents the Sbml data. And the second section is the tested model, the 3 model that we selected just now. And the third part is the best fitted parameter value. And this is the AIC table. So as you can see, here is the ranking table to show for the 3 different models. So from here, what we can see here is that the model 3 is being chosen as the best model with evidence of substantial support. Whereas for model 1 and 2, it shows that no support. No support means that they are actually significantly different from the model 3. So this section actually show the summaries of the everything. So basically the best model is 3 and the system type and also the best selected parameters. So all these information can be exported into an Excel file. So you just click on the export result and then all the file will be available in this zip file. So yeah, we can also visualize the fitting result. So here is the result for model 1, model 2, and model 3. So from here, what we can see here is that model 3 is the one that can actually well capture all these different expression trend. Yeah. So that's all for my the demonstration using the GUI. So currently we are still in progress of trying to improve the GUI by adding more features. But yeah, for now, I will pass to Russell to continue the demonstration using Python script.
All right, thank you, Jingwei. Yeah, I guess it's my turn to share the screen now. Okay, can you all see? Right. So thanks for sharing that with us, Jingwei. So for those of you who are more coding savvy, you may want to instead write your own Python scripts to use BMSS instead of the GUI, and that will give you a lot more flexibility in accessing BMSS functions, especially as we are still in the progress of adding new features to that GUI. For today's case study, I will also be using the same logic gate problem that Jingwei presented, and note that I'll be using screenshots of Jupyter for the demonstration. So first of all, I will go through how you choose and load models into Python using BMSS, followed by parameter estimation and model ranking. These are things that were covered in the GUI, and after that, I will go on to talk about some features that are not yet part of the GUI, such as identifiability analysis, storing and retrieving models in the database, sensitivity analysis, and finally, importing and exporting Sbml so that you can interoperate BMSS with other software that you are more familiar with. Okay, so first we need to load the models into BMSS, and we have the configuration files to do this. So Jingwei already gave you a preview of that. I just want to go into a little more detail here. So this is in the .ini or .ini format that's commonly used in many configuration files. And as you can see, we have here the mathematical topology of the model. We have the system type, and then we list down the states, parameters, inputs, and the equations. So once we have our files, we can iteratively call the from config function and load those models from the files into Python in the form of dictionaries, which contain all the fields that were in the configuration files. Then we have the experimental data, which we can combine with the models to perform parameter estimation. So when it comes to using BMSS, purely through Python, we do not enforce a particular data format, so you can write your own pre-processing function. I'm not going to go through that here, but I'll just say that the data will be in the form of dictionaries with containing the experimental data that needs to be fitted for each model. Once again, we have configuration files, which we will read using the getSamplerArgs function. So you can see on the right, we have a configuration file for the settings for curve fitting. And these contain stuff like information for numerical integration, such as the initial values. It also contains information such as parameter bounds and priors and initial guesses that are required for the curve fitting algorithm to work. So we combine the experimental data with the settings, with the models, and then we call our simulated annealing function to perform that curve fitting. So in this example, I'm using simulated annealing, but BMSS does provide wrappers for SciPy optimized package. So you can use like differential evolution or gradient descent, what have you. If it's on SciPy, then it should be on BMSS. Okay, so once we've run the curve fitting algorithm, we are returned a pandas data frame of sample parameter values from the algorithm. We can then use the calculate IC function to find the AIC and then rank the parameter sets and the models by the AIC value. BMSS does have the capability of calculating BIC and CAIC if you prefer, but the default is AIC. So the return value will be the data frame that you see at the bottom, which shows us that just as in the case of the GUI, model 3 is the one with substantial support. Right, so we have our AIC table, which tells us which model fits best in numbers, but we can also use the simulation capabilities of BMSS to plot this out and inspect it visually, as you can see here. Okay, and with this, I have covered the aspects that are available in the GUI. So I'm now going to move on to two types of analysis which were not part of the GUI. The first is identifiability analysis. So when we run a curve fitting algorithm, the sampler tries to converge in a neighborhood or distribution of the best parameter values. How do we know that this convergence has occurred? So BMSS has the plot steps function, which we can use, and it takes in the accepted values from the sampler, and then it generates a trace plot, which allows us to check the route that the sampler took, and then we can see that for some of the parameters, the sampler did indeed reach a stable distribution of values towards the end. Whereas for other parameters, such as SynMRNA3, convergence was not observed. So this means that the parameters were not actually successfully identified, probably because of correlation issues or noisy data. So this means that if we want to solve this, we can constrain the model more or collect more data or measure more state. For smaller models with fewer free parameters, we can actually perform identifiability analysis even before we run the experiment. So we've provided a Python wrapper for the strike-go algorithm developed by BioModel et al., which makes use of differential algebra. So once again, we have our configuration files, which contains information about the settings for the algorithm. After we read in those settings, we run the algorithm and we get our result, which is a dictionary that tells us which states are observable, which parameters will be identifiable. Right, and after we have, we fitted the models and gotten our best parameter set, we can then look into sensitivity analysis and see which parameters have the greatest impact on the performance of our genetic circuit, so that we know which parameters are good for tuning. So once again, we have the configuration file, so we read in that file, and then we now need to define an objective function related to the performance parameter that we want to optimize. So in this case, I'm going to define the performance as the concentration of peptide 3 at the end of the simulation. So I add this into the sensitivity arguments and then feed everything into the analyze function, and that gives me the result, which is generated using the salib library. We can then plot this result to get heat maps, as you can see. The one on the left is for first order sensitivity, and the center and right ones are for second order sensitivity. Dark values correspond to low sensitivity and light values correspond to high sensitivity. So if you look at the figure on the left, you can see that degradation of the peptide has a larger effect on the performance of the model, has a larger effect on the performance of the genetic circuit as compared to the synthesis of the mRNA. So if we want to tune our circuit, we may want to look more into degradation tags and whatnot, rather than the promoters for the mRNAs. Then moving on to the right, we have second order sensitivity, and we can see that certain pairs of parameters are more closely correlated with one another, such as the synthesis of peptide 1 and the degradation, whereas others are are more independent of one another. Okay, so next would be using the databases. The configuration files help us to define models dynamically, but in order to facilitate reuse, we have the database-driven approach by BMSS, and we can create new settings as a dictionary, and then we use the add to database function to add our favorite model, the one with the resource competition, and the relevant settings into the database. So the next time we try to do simulation or sensitivity analysis, we don't need to provide we don't need to write new configuration files from scratch, and we don't even need to reuse the configuration files. We can just pull them out of the database instead. So in this example, I use the quick search function to retrieve the model from the database, and the result is printed below. And you can see that the information here corresponds to what was in the original configuration file, except that I'm now pulling it out of a database and not a configuration file. All right, so this brings us to the last functionality, which is to import and export files to Sbml. So sometimes you'll want to work with things other than BMSS, so interoperability becomes important. We have the database to Sbml function. Sorry about that, Sbml function. And we type in the system type as well as the name of the settings, and BMSS converts all of that into Sbml files that we can use in other software. And we can also do the reverse, which is to take in an Sbml file and make a model out of it. So the result is printed below. You can see that this printed result is actually in the same format as the configuration file. Right. So this will allow us to work with stuff like iBioSim and Copasi. Okay, so that's all for the functionality. So useful info. BMSS is available on GitHub and PyPy, so please check it out. Let us know what you think, what you like and what you don't like. The preprint is also available on bioRxiv, so that you can check that out for a good summary of BMSS functions. And we also have a web page version of the database. And you can use this to see what models we have. And if there's something you want us to add, just drop us an email. This one's for the definition of the model itself. And then this one is for the parameters. So you can see things like the units and the bounds for parameter estimation, so on and so forth. And that brings us to the end of this presentation. Xingli and I would like to thank our colleagues from PoLab who have helped us a lot with the collection of experimental data. And we would also like to thank the many FYP and serious program students from our collaborators at the University of Pittsburgh who have contributed greatly to the development of this package. Finally, you can check out more of our work using the link at the bottom of this slide, EngBio, Sin City organization. And that's all. Timmy, do you have anything to say?
No, it's good. Yeah, thank you all.